This short video looks at cell division and a little bit on binary fission, but really focusing on mitosis, the kind of cell division that happens in eukaryotic cells. So just to give us some context here, there are really three types of cell division that are required in this course. One is an understanding of binary fission. Now, this happens in prokaryotic cells. It is a simple process of DNA replicating. The chromosomes, circular chromosomes, segregate, and cytokinesis is the process of one cell separating into two. That's been covered previously, and there's a fairly simple process that happens in prokaryotic, or often referred to as bacterial cells. Mitosis and meiosis are a type of cell division that happen in eukaryotic cells. Eukaryotic cells in multicellular organisms are considered diploid. That means that they have two homologous chromosomes for each of their chromosomes. So this small chromosome, there's two different versions of that, and the large one there's two different versions of that. For both these types of cell division, we start with S phase, which refers to synthesis of DNA, very much equivalent to the DNA replication that we see over here. What happens after the S phase is they either go into mitosis or meiosis, and those are different processes that sort those new chromosomes, those synthesized chromosomes, into different arrangements depending on what type of cell division is happening. And this is really the focus of this. Meiosis is part of sexual reproduction. As you can see here, we've got fertilization of gametes meeting to make a zygote. We discussed that in the organisms topic later on. So we're just going to focus on mitosis for today. So before we get into the details, we need to understand some terminology about how we refer to chromosomes. So again, here is a chromosome, here's the homologous chromosome. So say this is chromosome 8, you have two copies of chromosome 8 that are homologous, which means that they have the same genes in the same positions, but not the same DNA sequence. The reason for the difference is that one would be from your mother and one from your father. They're genetically different but have the same genes in the same locations, and so you have two copies of each gene. There's a position on the chromosome called a centromere, and this is used for attaching chromosomes to other chromosomes. When the DNA is replicated, all of the genes on this purple chromosome, all of this DNA is copied, and the same for all of the green one, and it remains attached to the other copy at the centromere. So now we have what we call sister chromatids. So the two purple sister chromatids are copied from each other with semi-conservative replication. We know that one strand of this is new and one is the original template, and same on the other side. So these are identical copies with the same genes in the same locations, but also the same sequence. So sister chromatids are identical copies, and of course there's two identical copies of the green one, whereas they still remain as homologous chromosomes. So in this case, on the left we'd say we have two chromosomes, on the right we have two chromosomes. On the left we have two chromatids, on the right we have four chromatids. So sometimes we can get confused as to what's a chromosome and what's a chromatid, so just bear that in mind. So the process of mitosis is basically just making sure that each of these chromatids gets separated and that one ends up in one cell and one ends up in the other cell, and the same needs to happen over here. So this process looks a bit like this. So we start off with 46 chromosomes represented by 46 chromatids. When those chromosomes are copied, we end up with 92 chromatids, which have to be separated out, half onto one cell, half onto the other. So interphase is the phase of the cell cycle where synthesis happens between those cell divisions, and then prophase, metaphase, anaphase, and telophase are the four phases of mitosis. These are each designed, as I said, to separate out the chromosomes. So prophase, there's a dissolution of the nuclear membrane, preparing those chromosomes to move around the cell, because normally they would be trapped inside that nucleus. At metaphase, they align across the division cleavage plane here, and as you can see, one sister chromatid on each side of that line, so that when we get to anaphase and they start to divide, they're being pulled by their centromeres using this spindle that's made of cytoskeleton. One chromatid goes one way, one chromatid goes the other, and in telophase new nucleuses form around each of those, and then cytokinesis, the step after, that cell will divide into two, and we end up with two cells with 46 chromatids each, representing 46 chromosomes. So Key thing about metaphase in mitosis is that they line up like this with one sister chromatid on each side, and the sister chromatids separate, so you have equal numbers of chromatids representing all of the chromosomes on each side.